Today's lecture focuses on yet another branch or facet of genetics that we will lump together as population genetics and evolution. Population genetics is the study of genetic variation within a population. So in order to understand that, we have to set a few definitions. A population is a localized group of individuals all belonging to the same species. Within that population, we can also refer to the gene pool, which is the total aggregate of genes in a population at any given time. For the most part, throughout the semester, we have focused on individual or single genes, and we've looked at individuals rather than groups. However, with population genetics and evolution, there is a switch to looking at genotypic and allelic frequencies in the population as a whole. So, as I just stated, we are going to focus on genotypic and allelic frequencies in a population for the remainder of this particular lecture. So let's start off with, first of all, describing how we can calculate genotypic frequencies. And that is calculated as the number of individuals possessing or having a genotype of interest divided by the total number of individuals in the sample. So in this very simple equation, the letter N designates the total number of individuals in the population, and the lowercase f indicates the frequency of each genotype. So you can see the very simple formula here for the three different genotypes that we know exist. Individuals in a typical dominant recessive relationship can either be homozygous dominant, heterozygous, or homozygous recessive. And we can calculate genotypic frequencies given the number of individuals of a specific genotype and the total number of individuals in the population. So if we look at the frequency of the homozygous dominant genotype, that is the number of homozygous dominant individuals divided by the total number of individuals in the population. And you can see the formula holds true for the frequency of the heterozygous genotype as well as the homozygous recessive genotype. So let's look at a specific example and how we can calculate the genotypic frequencies. In this very simple example, we are looking at a fictitious wildflower population where there are only two alleles for flower color given in this population. Those two alleles are listed here for you as the dominant being red and the recessive being white. This is a classic dominant recessive relationship based on concepts we talked about at the beginning of the semester. We know that in a classic dominant recessive relationship and the allele designations I just went over, the possible phenotypes in this population are red flowers and white flowers. And given the classic dominant recessive relationship, we can also discuss or label the genotypes. If this is a classic dominant recessive relationship and red is dominant to white, individuals with the red phenotype can either be homozygous dominant or heterozygous, and individuals with the white phenotype are homozygous recessive. Now, in this example, in this fictitious wildflower population, I'm going to give you some data and say that in this population, there are 200 plants that are homozygous dominant, 160 plants that are heterozygous, and 20 plants that are homozygous recessive. It's not too difficult math to just add that up and determine that that means there are 500 plants total in this population. Based off of the formula in the previous slide, we can very easily calculate the genotypic frequency. If we know the number of individuals with a given genotype, and we know the total number of individuals in the population, it's a simple division problem to calculate the genotypic frequency. So for example, for the homozygous dominant genotype, there are 200 individuals that have that genotype. Divide 200 by 500, which is the total number of plants. That gives you 0.64, which is the genotypic frequency for homozygous dominant. And you can do the exact same calculation for the heterozygous individuals and the homozygous recessive individuals. As stated at the beginning of the lecture, in population genetics, we can not only calculate genotypic frequencies, but we can also calculate allelic frequencies. This too is a very straightforward calculation as long as you know the number of copies of a particular allele in a sample population divided by the total number of alleles in the population. And you can see the very simple formula listed for you here. Again, the frequency of an allele is the copies of that allele in the population divided by the total number of all alleles for that particular trait in a population. So let's go back to the wildflower example. In the wildflower example, we can calculate the allelic frequency for the dominant red allele as well as for the recessive white allele. For the dominant red allele, there were 320 individuals that were homozygous dominant. But in that genotype, there are 2 copies of the dominant allele. So we would take 320 and multiply that by 2 to represent the total number of dominant alleles present in the homozygous dominant individuals. Plus, we have to add to that the number of dominant alleles represented by heterozygous individuals, which was 160. This is then equal to 800 divided by the total number of alleles in the population, which would be 1,000. Going back to that example again, if there were 500 individual plants, that means that there must be twice as many alleles for flower color in the population because every individual has 2 copies. So again, the calculation is 800 divided by 1,000, which is 0.8. We can use that exact same calculation for the allelic frequency of the recessive white allele. In the homozygous recessive individuals, there were 20, again, that were homozygous recessive. And there are 2 copies of that recessive allele present in each of those individuals. So we would multiply 20 times 2. We also have to account for the recessive alleles present in heterozygous individuals, which would be 160, which is a total of 200 recessive white alleles divided by 1,000 total alleles in the population for flower color, which is equal to 0.2. So what we've just calculated on the previous slide is the allelic frequency for the red and the white allele in this fictitious wildflower population. We calculated 0.8 as the frequency of the dominant allele, which essentially means that 80% of the flower color genes have the dominant allele in the flower population. We also calculated the recessive allele frequency as 0.2, which also means that 20% of the genes found in this particular population are represented by the white or recessive allele. If we continue along those lines and we make the assumption that we have a non-evolving population, we can calculate the 3 possible genotypes into the next generation. And the way that we can do that is using our old friends, the product law and the rule of addition. So hopefully you can go back and review some of that so that it's really clear in what these calculations really mean. Now, I mentioned that we are going to make the assumption that this is a non-evolving population. And the second half of this lecture will look at some of those assumptions and tie in just a little bit of what it means to be a non-evolving population. So we will get to that in just a little bit. So using the allelic frequencies that we just calculated, 0.8 for the dominant allele and 0.2 for the recessive allele, we can again use the product law to predict the probability of individuals being homozygous dominant in the next generation. What is that probability of receiving two dominant alleles? It is the product of the individual probability. So if the individual probability of receiving a dominant allele is 0.8, we multiply that by the probability of receiving another dominant allele, 0.8, which gives us 0.64. So 0.64 is the probability of being homozygous dominant in the next generation based off of the calculations we just made. We can also use that same logic for calculating the probability of individuals that will be homozygous recessive in the next generation. That too applies the product law, which is the product of the individual probabilities. So if the probability of receiving one recessive allele is 0.2, multiply that times the probability of receiving another recessive allele, 0.2, which gives us 0.04. This is the probability of individuals that will be homozygous recessive in the next generation. Now, calculating heterozygous individuals is going to require a little bit of finesse as well as recalling the rule of addition. We know that there are two different ways individuals can be heterozygous. They can receive the dominant allele from the first parent and the recessive allele from the second, or vice versa. They can receive the recessive allele from the first parent and the dominant from the second. What is the probability of receiving one dominant allele and one recessive allele in that order? 0.8 times 0.2, which is 0.16. And the alternative, the other way an individual can be heterozygous, receiving the recessive allele from the first parent and the dominant allele from the second, 0.2 times 0.8, which is 0.16. When we consider that there are more than one way that individuals can be heterozygous, we have to add up those individual probabilities. So that is 0.16 plus 0.16, which is 0.32. And that is the probability of being heterozygous in the next generation based off of the calculations we have utilized. Based on that very straightforward mathematical model for genotypic frequencies of P plus Q equals 1, we can expand that for allelic frequencies. For allelic frequencies, the probability of receiving two dominant alleles, as we stated in one of the previous slides, is 0.8 times 0.8, which is 0.64. If we utilize our P designation as letting the allelic frequency of the dominant allele equal P, this then is P times P, which is also P squared. We can hold that same logic through for homozygous recessive genotype. The probability of receiving two recessive alleles is 0.2 times 0.2, or Q times Q, which is also referred to as Q squared. And the probability of heterozygous individuals is either the dominant first and the recessive second, or the recessive first and the dominant second. 0.8 times 0.2 plus 0.8 times 0.2. This can also be rewritten with the substitution of P and Q for the dominant and the recessive alleles, which could be simplified as 2PQ. So with the Hardy-Weinberg law, we can expand it again to include allelic frequencies. And that formula is P squared plus 2PQ plus Q squared is equal to 1. And these are the two basic formula that we will utilize for Hardy-Weinberg. One for genotypic frequencies and one for allelic frequencies. So this slide just shows you another view as how the allelic frequencies can be utilized to calculate genotypic frequencies. So you can utilize a simple Punnett square using those frequency calculations that we've already assigned, P and Q, to determine what the frequency of homozygous dominant individuals is, which is P squared, the frequency of homozygous recessive individuals, which is Q squared, and the frequency of heterozygous individuals, which is 2PQ. Now, this formula can also be expanded for more than just two alleles. However, for time purposes and for ease of instruction, we are going to stick with the two allele model for example calculations. So let's move on to an example of how Hardy-Weinberg can be utilized to estimate the percentage of the human population that carries the allele for a particular inherited disorder. In this particular case, we are talking about a disorder known as PKU, which is phenylketonuria. It is the inability to metabolize the amino acid phenylalanine, and it is an autosomal recessive disorder. It turns out that about 1 out of 10,000 individuals have PKU. So given that information, what we know is the frequency of individuals that are homozygous recessive. Because this is an autosomal recessive disorder, we know that individuals that will have the disorder have to have the genotype being homozygous recessive. And using our formula, we know that the frequency of individuals that is homozygous recessive is equal to Q squared. This is information that we can utilize that's been given to us. So Q squared then is equal to 1 out of 10,000 because stated above we have that frequency that's already been established. 1 out of 10,000 can be rewritten as 0.0001. That is equivalent to the Q squared value. What we need to do is solve for Q, which would then be to take the square root of that value, which then is 0.01. So the frequency of the recessive allele in the population is 0.01. Given our Hardy-Weinberg equation, we can solve for the frequency of the dominant allele designated by the letter P, which would then be P is equal to 1 minus Q, using that simple algebraic conversion that I mentioned just a minute ago. So 1 minus 0.01 is equal to 0.99. That gives us the frequency of the dominant allele in the population. So now we've established what the frequency of the dominant allele is and the frequency of the recessive allele. However, in this question, what we're interested in is the number of carriers of the allele for a particular disorder. And a carrier would be the individuals that are heterozygous. They are carriers because this is the classic dominant recessive relationship, and they would have the dominant phenotype, which would be they would have a normal ability to metabolize phenylalanine, but they do have the potential of passing on a recessive allele to their offspring. So how do we calculate the frequency of carriers? That is the frequency of heterozygous individuals in the population, which would then be 2PQ. Since we've already calculated P and Q, we can simply plug in those numbers to calculate 2PQ, which is 0.0198. Approximately 2% of the U.S. population, therefore, carries the PKU allele. Now, in nature, it turns out that populations are dynamic. They are in constant flux. And what we're going to discuss then are factors that can alter allelic frequencies in a population. In addition to that, these next concepts also tie in with some basic evolutionary concepts as well. One of the factors that I mentioned a few slides back was that in order to utilize the Hardy-Weinberg law, we had to make some assumptions. We made the assumption that population size was going to be very large, that there was no migration occurring in this population, there was no net effect of mutations on this population. We had to make the assumption that mating was occurring randomly and that no natural selection was taking place. So again, what we're going to do is look at these parameters to see how these may alter allelic frequencies in a population. The first assumption is that the population size is very large. If the population size is not large, we can see things that occur such as genetic drift, which may be caused by what's known as the founder effect or bottleneck effect, and we'll look at an example of that in just a minute. So let's look at the effect of not having a large population size. This is referred to as genetic drift and is a change in the gene pool in a small population. Now, this is something that occurs to random chance. In this example, we are again looking at a wildflower population with red and white flowers in the far left of the slide. If only 5 of those plants continue on to produce offspring in the next generation, we see some altered patterns that might occur. And then in this example, which is in the center of the slide, if only 2 of those plants continue on to produce or allow for production of offspring in the next generation, what has happened in this very small population is that we've entirely lost the white flower color overall, as well as the recessive allele. And again, this is a very specific example, but it does drive home the idea of what happens or what can in fact happen in small populations. This is now all due to random chance, in that if we have only certain individuals that can reproduce to produce offspring in the next generation, and that number gets even smaller, we can see how we can potentially lose an allele in a population. Now, this effect can also be seen in what's known as a population bottleneck. This is a very simple, straightforward evolutionary concept of genetic drift, and what it does is result in a drastic reduction in population size. In this example, you have all of these individuals represented by the balls or the spheres in the bottle. And if only a few of those individuals are allowed to move into a new situation, either surviving some type of bottlenecking event, you now have lost some of the variation in that original population because of the individuals that are allowed or have survived into the next generation. So here too you can see an example of how allelic frequencies can change dynamically based on a situation like a bottlenecking event. For those of you that might be more mathematically inclined, this last figure includes a table that shows what could happen in a very small population. If you have a very small population that is reproducing into the next generation, given the genotypic makeup of those individuals, you can then predict what's going to happen to allelic frequency. So for example, the very first row of the slide or the table, you can see the 2 genotypes that are producing offspring are both homozygous dominant. In this particular case, we've completely lost the recessive allele in the next generation because of the very small sample size, 2 individuals in this particular case. So it just drives the point home, I think, a little bit more strongly. The next assumption that must be maintained in order to utilize the Hardy-Weinberg equation is that there is no migration occurring between populations. The effect of migration on allelic frequencies can be dramatic, And so we are going to make the assumption that no migration is taking place. So in this example, what you can see is the overall effect of migration when it does in fact occur. In this graph, what you're looking at is the frequency of a given allele in various human populations. That allele is designated as FY null 1, and what you can see is that it is not present in African individuals. It is completely 100% present in European individuals. And in European Americans, we begin to see a slight shift overall in loss of that particular allele, in that not every single individual of European American descent will have that allele. And then right in the middle, you see the individuals represented by the blue bars, which are African Americans. What you are seeing here is that, again, in the original African population, this allele is not present at all. But in African American individuals, you begin to see the presence of the allele appear. This is due, again, to migration between different populations of individuals. In order to utilize the Hardy-Weinberg equation, we must make the assumption that there is no net mutation occurring in this population. The effect of mutation on allelic frequencies is extremely minimal. We discussed previously that mutations are processes that produce alterations in DNA or chromosome structure. Now, we can actually calculate mutation rates as the number of new mutant alleles divided by the given number of gametes. It turns out that the mutation rate tends to be very, very small, and the impact of mutation on allelic frequency remains very small as well. This is just another view of how mutation might affect evolutionary processes and why we have to assume overall that no net mutation is taking place. If you look at the top left-hand corner, you can see the initial allele frequency in this population, the dominant allele represented by 0.9 and the recessive allele represented by 0.1. Again, allelic frequencies in this particular population. If we then utilize that information to determine the genotypic composition, we can then calculate genotypic frequencies based on those allelic frequencies, and you see that in the far right-hand corner. If we then utilize that information as far as carrying on to the next generation, again, we can use that information to make the prediction about genotypic frequencies. However, if there are, in fact, mutations that occur, those mutations can have an effect on allelic frequency. So, in the final generation, we see that there is a shift in the allelic frequencies ever so slightly from the initial frequency. And again, this is ever so slight, so mutation traditionally is considered a weak mechanism of evolution. The Hardy-Weinberg law also takes into consideration or makes the assumption that random mating is in fact taking place. If non-random mating occurs, we can see things known as inbreeding depression. This would be a situation, inbreeding, where there's a mating between relatives, and this increases the proportion of homozygous individuals in a population for a given allele. The consequence of inbreeding is inbreeding depression, and this is a measure of loss of fitness. Oftentimes in these situations, unmasking of potentially deleterious recessive traits can oftentimes take place. So what we see in this particular table in the center column is that the frequency of heterozygous individuals continues to decrease when we have non-random mating situations. Lastly, Hardy-Weinberg makes the assumption that there is no natural selection taking place. With natural selection, what we have is a differential success in the reproduction of different genotypes. What that really means is that certain genotypes might have an advantage in survival or in reproduction overall. We can see the effect of natural selection overall on allelic frequencies in the next slide. So again, in the left-hand panel of this figure, you can see the initial allelic frequency in this particular population. And we continue on to be able to calculate the genotypic frequency, which is shown in the right-hand corner of the figure. However, when natural selection occurs, again, certain genotypes will have an advantage in survival or in reproduction. And so in this example, what we're seeing is that some of the individuals that are heterozygous die and cannot move on to reproduce in the next generation, as well as individuals that are homozygous, recessive in this particular case, do not survive in order to reproduce. That then changes the composition of the allelic and genotypic frequency in the population, specifically those that have the ability to reproduce. And because of that, we see alterations in the final allelic frequency because not all of the individuals survive in order to reproduce and carry on that allelic information to the next generation. In this last slide, you can see a little more obviously what is happening when natural selection does in fact take place. In this example, we are looking at the selection scheme, meaning how strong or weak is the selection process. If you have a very strong selective component that is depicted by the red, you can see that this can in fact have dramatic effects in allelic and genotypic composition. The opposite end of that is true if the selection process is weak, represented by the blue. There's very little changes in the surviving genotypes that we see.